Good morning, students. Today I'll be doing with chapter 11, Biotechnology, Principles and Process. So let's understand what is biotechnology. How do you define biotechnology? If I break it into two terms, then it's bio and technology. So technology used in biological processes, that is biotechnology. Now, biotechnology, that means biotechnology deals with techniques of using live organism or enzymes from organism to produce products and processes useful for humans. Now, if we think in this sense, even making of curd, bread, and wine, which all include microbes, it's a microbe-mediated processes. So all these things can be thought of as biotechnology. But in today's scenario, biotechnology is used in a restricted sense. It refers to such of those processes which use genetically modified organisms to achieve the same on a large scale. There are further many more processes or techniques that are included under biotechnology. For example, you can see on this slide also, for example, in vitro fertilization, that is test tube baby, synthesizing and using a gene, then preparation of DNA vaccine and correcting a defective gene. Now, according to European Federation of Biotechnology, EFB, according to them, they have defined biotechnology as techniques of using live organism or their enzymes for products and process useful to humans. Now, what are the core principle techniques, core techniques in modern biotechnology? What are the core techniques? There are two core techniques. There are many, but among that, there are two core techniques that enable the birth of modern biotechnology. One is genetic engineering. The first one is the genetic engineering, and the second one is bioprocess engineering. Genetic engineering, it is the technique in which the genetic material, that is the DNA and RNA, it is chemically altered and introduced into the host organism to change the phenotype. Now, the DNA is taken, or RNA is taken, it is genetically, alteration is done in that, and obviously when you introduce into the host, the phenotype of the host is changed. The second core technique is bioprocess engineering, that is maintenance of sterile ambience in chemical engineering process for growing desired microbe or eukaryotic cell in large quantities for the manufacture of antibiotic, vaccines, and enzymes. As just now I have told you that in the process of biotechnology, we produce enzyme, vaccines, antibiotics. So these are being produced through microbes or by a eukaryotic cell. So these eukaryotic cells or microbes need to multiply or grow, and they require a sterile ambience. So maintenance of sterile ambience is also one of the core techniques of biotechnology. Now, what are the basic steps in producing genetically modified organism? There are three basic steps in producing it. One is identification of DNA with desirable gene. Second is introduction of this identified DNA into the host. So first you have to identify the DNA with desirable gene. कौन सा desirable gene आपको चाहिए? Second, you have to introduce this DNA into the host, and the third basic step of producing a GMO is maintenance of introduced DNA in the host and transfer the DNA into its progeny. That DNA which we have introduced into the host should transfer into the progeny. So these are the three basic steps of genetically modified organism. The very first step is what? Identification of DNA with desirable gene. Now, if we talk about this first step, then so the traditional hybridization procedures which were used in plants and animal breeding very often led to what? These traditional hybridization was also a type of biotechnology, but in this traditional hybridization process, there was a big disadvantage. That is, with the desired gene, undesirable genes were also transferred. And in this genetic engineering, what are we doing? Only desirable genes are introduced. The undesirable gene does not get transferred. And when we are transferring the desirable gene, then we are creating a recombinant DNA. The second step is introduction of this identified DNA into the host. In this, we require a vector DNA, such as a plasmid, is used to deliver this alien piece of DNA into the host organism. You can see over here, what are plasmid? These are autonomously replicated DNAs present in circular DNAs present in bacteria. We introduce a piece of our DNA, desired gene, containing desired gene into the plasmid. And then, see, you can see over here, suppose this is the desired gene. Now you call it as a recombinant plasmid, and then you introduce this recombinant plasmid into the host. Third is maintenance of introduced DNA in the host and transfer the DNA into its progeny. Now this piece of alien DNA it has to multiply. So we put this piece of alien DNA near ori. You have studied what is ori in chapter 6. That is origin of replication. So that it gets replicated, because it cannot multiply on itself. So we put it near the ori so that it can multiply, and then it has this alien piece of DNA. It becomes a part of the chromosome, which has the ability to replicate. And in this chromosome, we all know that there is a specific DNA sequence known as ori, which is responsible for initiating the replication. Therefore, for multiplication of any alien piece of DNA in an organism, it needs to be a part of the chromosome, and it has to be present in near the ori. Thus, the alien piece of DNA is linked with the ori, so that this alien piece of DNA can replicate and multiply itself in the host organism. And what do you call it? You call this as cloning. So these are the three basic steps. Now, let's see how the first recombinant DNA being formed. This first recombinant DNA was constructed by linking the piece of DNA, by linking the piece of this gene encoding antibiotic resistance with the native plasmid. As I told you just now, these plasmids are autonomously replicated circular extra chromosomal DNA. And this native plasmid was of Salmonella typhimurium. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistant gene by cutting out the piece of DNA from the plasmid, which was responsible for conferring antibiotic resistance. This cutting of DNA at specific location became possible with the discovery of the so-called molecular scissors, which is now known as restriction enzyme. Now, this cut piece of DNA was then linked with the plasmid DNA. And this plasmid DNA now act as vectors to transfer the piece of DNA attached to it. The enzyme ligase, it joins the ends of the DNA molecule. This makes the new combination of circular autonomously replicating DNA, and it is created in vitro, and now it is known as recombinant DNA. Now, when this DNA is transferred into E. coli, which is bacteria very closely related to Salmonella, it could replicate using the new host DNA polymerase enzyme and make multiple copies of it. So, This is how the first recombinant DNA was produced. Thank you. And the further topics will be coming in the further videos.